Okay. Thank you everyone for coming. Welcome to this second episode. Is this working, Nicole?
It is.
It is? Okay. You laughing at me?
No, it's projecting on my computer.
Oh, okay. I can hear my voice there a little bit. Our guest today is Dr. Karie Altman, straight from St. Bonaventure down the road, west of us. Karie got an undergraduate degree in biology from Bonaventure University in Michigan, followed by a PhD from Altman University in Michigan as well, and has postdoc experience from University of Pittsburgh after that, more recently, and then she's in her fourth year. Did I get that right? Yeah, as assistant professor in the biology department at St. Bonaventure University. And I'm always very excited to hear about amphibians, so I'm very excited about this. So I'm gonna give this to you.
Okay. Thank you for that introduction, and thank you for having me today. I'm excited to be here. My students are also excited that I'm here because I had to cancel a class today. So today I'm going to talk a little bit about the temperature dependence of amphibian disease. But I'm also going to talk more about just kind of threats to amphibians in general and what I've been studying over the last several years. So to get started, organisms often face several threats simultaneously. So with this example, in looking at amphibians, the one thing that I often focus on with amphibians is their exposure to disease, but that's not the only threat they're facing. They're facing things like pollution, habitat fragmentation, invasive species, and one that I focus on largely has been climate change, and that's where the temperature dependence comes into play. So when thinking about how I might structure this talk, I was looking for a little bit of inspiration. And I wish I could say I went to like one of our finest researchers of our day, but I went to Taylor Swift. So the way that I've kind of structured this is I'm going to take you through sort of my academic eras and how amphibian research has been involved in each of them. I'm glad that you chuckled a little bit because coming into somewhere where I don't know anyone, I was like, ooh, is this gonna land? So thank you. So what we're going to be doing today is just kind of going through a little bit of my research from each of my academic stages. We're going to start with my grad school in Michigan, walk through my postdoc, and then a little bit about what I'm doing at St. Bonaventure. So to get started with what I really focused on at Oakland, let me. So all of these we're going to look at amphibian disease, but we're going to look at different diseases. All right. Initially, we're going to start with trematodes, then we'll get into the chytrid fungus, which I'll talk more about. And then we'll look at how disease manifests with other threats. So things like climate change, as well as pollution. All right, so climate change is really multifaceted. There's so many aspects of it. We think about carbon emissions, we think about melting polar ice caps, more extreme weather events. And where my focus in climate change is how is temperature going to impact species interactions? So in thinking about this, the first thing I want to introduce is this concept of a thermal performance curve. All right, so these show how an organism performs over a range of temperatures. So we're going to have our temperature on the x-axis, some aspect of an organism's performance on the y-axis. It might be something like swimming speed or resistance to a parasite. And what we see is that organisms' performance typically increases with temperature over a range until it reaches an optimum, and then performance typically drops off pretty quickly beyond that. There we go. All right. So when we talk about temperature, as the climate changes, temperatures are certainly predicted to increase in mean. All right, we've heard global warming, right? But it's not just that. In addition, temperatures are predicted to increase in variability around that mean, so getting more and more variation. And so that variation will likely impact organisms and impact species interactions. And I apologize, I cannot get this to get rid of, there we go, okay, woo. All right. So when we think about how these changing temperatures are going to affect organisms, something we want to keep in mind is this idea of acclimation, all right? So acclimation is just an organism's ability to adjust its physiology in response to any environmental change. But for today, we'll focus on an environmental change in temperature, all right? So there are a number of ways that acclimation can impact organisms. I think the most straightforward one to understand is called the beneficial acclimation hypothesis. And this basically says that organisms acclimated to a particular temperature will perform better at that temperature relative to unacclimated organisms. So for an example of this, we'll take a look at these data showing burst speed in tadpoles, all right? And here we had several test temperatures, and tadpoles were acclimated to either a cooler temperature, blue, that's 15 degrees in blue, or a warmer temperature, that's 25 degrees, and that's shown in red. So here, what we see is that at cold test temperatures, our cold acclimated tadpoles are swimming faster than our warm acclimated tadpoles, but then the opposite is happening at high temperatures. So this is basically saying whatever conditions you're used to, you're going to perform better at those conditions than an organism that was just switched into those conditions. Now, in thinking about this in how it relates to a species interaction, such as a disease, now we've got not only the temperature dependence of an infection shown in black, but temperature can have different effects on the host and the parasite, the disease-causing organism. So it's really important, like when we're trying to predict how temperature will impact an infection, we have to consider temperature impacts on both the parasite, represented here in red, and the host, represented here in blue. But it's hard to do that often because hosts and parasites are often experiencing the same shifts in temperature. So we'll account for that in our experimental design that's coming up. So in terms of studying effects of temperature variation on disease, amphibian parasites are a really great study system for that. Both organisms in this system are ectotherms, so their body temperatures are going to be fluctuating with the environment. But additionally, everyone here, I've heard some excitement about amphibians, and that's awesome. But oftentimes when I present to like molecular and cell biologists, they're like, why do I care about frogs? So I will also say that we have a lot of ectothermic human parasites or human vectors of parasites. So things like mosquitoes, ticks, snails, which harbor trematodes like schistosomes. So having a general understanding of how temperature variation impacts parasites, even though we're doing it in amphibians, I think it's a worthwhile pursuit. All right, so in the past, studies of thermal acclimation on amphibian susceptibility to parasites has generally supported that beneficial acclimation hypothesis that I talked about. So what we're looking at here are results from a study where they infected Cuban tree frogs with a fungal parasite. And here we have a couple treatments. We have cold acclimated frogs in blue, warm acclimated frogs in red. This left column here, we see that these frogs were just kept at the same temperature the entire experiment. And then this column, this shifted column, the cold acclimated frogs were moved to warm temperatures. The warm acclimated frogs were moved to cold temperatures. And we're looking at the intensity of infection on our y-axis. So what we see here is that for warm acclimated frogs, it didn't matter whether they experienced a temperature shift. However, for cold acclimated frogs, they experienced higher infection levels following that temperature shift. We're going to stay in the same study, the same parasite system, a fungal pathogen. You'll read the graph the same way, cold acclimated in blue, warm acclimated in red. But here we see that no matter what temperature amphibians were acclimated to, following a shift, they had higher pathogen loads. All right. Now, that's great, but what I showed you initially was that thermal performance curve. All right. And so if we only include 2 performance temperatures, the results that we get depend on what performance temperatures we choose. All right. So we might see a decline in performance with an increase in performance temperature, but we have this whole thermal performance curve that's in the background. And so based on what study temperatures we choose, we might get very different effects. So one thing I wanted to do was start testing more of the temperature range for these amphibians. Now, additionally, prior to what I started doing, we had done most of the work with acclimation using only 2 acclimation temperatures. And it's possible that just like here, where we saw this nonlinear effect of performance temperature, maybe there's a nonlinear effect of acclimation temperature. All right. And so what that would look like in a graph would be, let's say we've got performance temperature on the y-axis, whatever level of performance, or sorry, on our x-axis, whatever level of performance on our y-axis. A nonlinear effect of acclimation temperature would look something like this, where our cold and warm acclimated organisms are grouping together in terms of their performance, and then intermediate acclimated organisms would be somewhere else. Here I'm showing below, but it's possible that they could outperform our extreme acclimated organisms. So what we do in these sorts of acclimation studies is we maintain our study organisms at a certain acclimation temperature. Here I'm showing 3, and then we'll switch them to a new performance temperature. So some acclimated to our low temperatures will stay there. Some will go to a new higher intermediate temperature. Some will go to an even higher temperature. All right. And we'll do that across the board. So what we'll focus on for this study is an amphibian parasite. Oop, nope, not yet. We're going to focus on research questions first. All right. So here we're looking at how tadpole acclimation temperature influences its susceptibility to Ribberroia infection, and does temperature have the same effects on all stages of Ribberroia infection? Now is a great time to introduce our parasite. So Ribberroia ondatre, it's a trematode parasite, and it's got this really cool complex life cycle where let's start in our definitive host. That's where the adult worms live. They will release eggs in their feces, and the stage of the parasite will then go into an aquatic snail. That snail will release cercariae. That's the parasite stage. Those cercariae will then swim around in the water, find an amphibian intermediate host, and kind of burrow into the skin there. As they do, they'll lose their tail and encyst as metacercariae. All right. Now, the person who drew this diagram, it wasn't me, but whoever it was, they knew what a frog looked like. All right. This parasite is known to cause limb malformations and sometimes extra limbs within amphibians if it gets in at the right stage of development. And although that's cool, it's not what I studied. So this infection process, the part that I want to focus on for this talk are the cercariae getting into the frogs and encysting as metacercariae. All right. So I want you to imagine that this is a cross section of amphibian skin. And first, that cercaria needs to encyst. It needs to get in, burrow in, lose its tail, and become a metacercaria within that tadpole. All right. But then once it's there, it's got to survive, and it's got to survive until that amphibian intermediate host can be consumed by the next host in the life cycle. So we're going to explore how acclimation is affecting each of these steps. All right, so we had a design similar to what I already introduced, all right, except instead of only 3 temperatures, we crossed 6 acclimation temperatures with 6 performance temperatures, all right? Following that temperature switch, we exposed each tadpole to 20 Riberoia cercariae. Remember, cercariae are these little swimming stages of the parasite. Now, remember I mentioned that often, not often, always in nature, the hosts and the parasites are experiencing the same temperatures. And so it's hard to tell whether we're looking at an acclimation effect on the host or on the parasite. And so this system offered a really cool way to kind of tease those apart, because the cercariae are released from snails. They're released from a different host. And so what we could do is we acclimated the parasites to the performance temperature. So our parasites in this experiment, they never experienced a temperature change, only our host tadpoles did. So if we find any sort of acclimation effect, we know it's an effect on the host. All right. Following that, we were then able to track parasites through time, and I'll show you how we did that. All right, so the first thing that we looked at was, how does thermal acclimation affect the proportion of parasites that were successfully able to encyst within that tadpole? And the way that we did that is, before exposing the tadpole to the cercariae, we tagged them with a fluorescent dye so that we could find them really nice and easily. So you can see here, like, this is the section of the tadpole that we're looking at. And you can see every glowing green dot is a metacercaria. So we measured the proportion of encysted parasites by just dividing the number of metacercariae that we found by 20, which is how many we exposed the tadpole to. All right. So what we found in this graph, we have performance temperature on our x-axis. Our measure of performance is the proportion encysted. That's on our y-axis. And although we acclimated each tadpole to just one temperature, I figured you wouldn't want to look at 6 different curves. So I've grouped things according to our 3 coolest acclimation temperatures in blue, our 3 warmest are in red. And so what we see here is consistent with what we would expect for beneficial acclimation. We see that our cool acclimated tadpoles had fewer encysted parasites at cool temperatures relative to the warm ones, but that pattern is reversed at higher temperatures. Here, our warm acclimated tadpoles are outperforming our cool one. Because remember, performance here, better performance would be fewer parasites in the tadpole's eyes. All right. So with encystment, we saw results consistent with beneficial acclimation. The next thing we did was, remember I said that once those parasites encyst, they have to survive there. And so the second measure of tadpole performance was their ability to clear these metacercariae, clear these parasites. And we did that by, again, I'm showing you this region of the tadpole. We did that by taking photos of the tadpole 12 hours post-exposure, and again, a week later. And so what we did was we could actually count, okay, this is in the same tadpole. How many parasites did they have initially? How many parasites do they have after a week? And so you can see here that, like, up here, this one happened to have 4. You can see that numbers 1 and 4 have been cleared over the course of the week, indicating the tadpole is able to get rid of those. And that's a good indicator of their performance. All right, so what we're looking at here is, again, performance temperature on the y-axis. This time, our measure of performance is the proportion of metacercariae that tadpole was able to clear. So higher proportion cleared equals better for the tadpole. And this time, I divided things into 3 acclimation temperature groupings: our lowest 2 temperatures in blue, our intermediate temperatures in purple, and our highest acclimation temperature in red. And what we see here, it's not beneficial acclimation, all right? It kind of was at low temperatures, it kind of was at higher temperatures. We did have our highest acclimated group performing pretty well there, but actually across the board, it was our intermediate tadpoles that did the best. All right, so that explanation was not quite as simple. So what we saw here was that thermal acclimation effects on green frog tadpole susceptibility to Riberoia depended on the stage of infection. When it came to encystment, that pretty clearly supported beneficial acclimation. However, when it came to clearance of those metacercariae, there might have been a hint of beneficial acclimation at the warmer temperatures, but it seemed like maybe those cold acclimated tadpoles were going into maybe some sort of dormancy and had really depleted performance overall, even at temperatures that they should have been used to. All right, so we'll move on next. That's trematodes and climate change. All right. Next, we're going to stick with this idea of climate change as a threat to amphibians, but now we're going to switch our disease system. We're going to look at a different aspect of climate change, and we're going to look at a different disease. So Batrachopytrium dendrobatidis, that's a mouthful. We're going to call it BD. This is a fungal pathogen that has caused declines in a lot of amphibian species worldwide. You can see that across the world, especially in like Central America, we have a lot of declining species. Many of the species in North America seem to be doing just fine. That doesn't mean they don't get BD, they definitely do, but the declines have been much less widespread. So this is a big deal in terms of pathogens. When I first started my research, I would explain to people that amphibians are like in this pandemic, this fungal pandemic. And that word pandemic did not have much meaning back then, but now people seem to get it, that like, ooh, this is a big deal. All right, so keep that in mind. We'll return to BD in a little bit. But first, I want to remind you that amphibians go from tadpoles in the water, they'll metamorphose and come on land as frogs. And the amount of time that an amphibian spends in the water, it's plastic. It can change depending on environmental characteristics like density, whether there are natural enemies present in that water, as well as hydroperiod. All right, so a lot of amphibians are laying their eggs in vernal pools that are going to dry up throughout the summer. And so with that, what we expect is that with climate change, some of those ponds are going to dry out faster. All right, so that's kind of my link to climate change here. Now, regardless of how much time they spend in the water, typically bigger is better for these amphibians. The bigger they can get, the bigger a size they can emerge at, that kind of gives them a better chance at doing well once they metamorphose. All right, so this time we're sticking with climate impacts, we're sticking with disease, but different ones. All right, so now our focal pathogen is going to be BD. All right, and then our climate impact, we're focusing on the fact that warming temperatures could increase the rate at which ponds dry. And we're going to study this using northern leopard frogs. All right, so a couple predictions to get us started. As we increase the rate of pond drying, that should increase tadpole developmental rate. All right, if you are a tadpole and your choice is develop fast or die as the pond dries out, you're probably going to increase development. All right, so we expect developmental rate to increase. We expect tadpoles to metamorphose sooner when they experience drying. All right. However, we also expect that will come at the cost of growth rate. All right. Their growth rate in the larval period will likely decrease. They'll metamorphose at smaller sizes. The other thing we wanted to explore with this study is what then happens after metamorphosis. All right, can pond drying have carryover effects into post-metamorphic life? And the things we explored here were the frog's susceptibility to BD infection, as well as post-metamorphic survival. And we expected frogs to be more susceptible if they're experiencing drying as tadpoles, and to have reduced post-metamorphic survival if they experienced drying in the larval environment. So the way that we tested this, we put 40 northern leopard frog tadpoles into mesocosms outside. This was done at University of Pittsburgh's Pymatuning Laboratory of Ecology. And we raised them under 3 drying treatments. So we've got time on our x-axis, the water depth over time. And by drying treatment, I mean my undergrads and I were out there like bucketing out water every week. So over time, we have these 3 treatments. Some of the pools were kept at a constant water level. Some were dried at a medium rate, and some were dried very quickly. And then we just kind of left a very small amount of water at the end once we got down to kind of the bottom of our tanks to allow for them to metamorphose. So what we did was we characterized some developmental effects as these tadpoles were coming out of the mesocosms and metamorphosing. But then once our frogs metamorphosed, we brought them into the lab, and now they were kept at uniform lab conditions. And then we measured 2 potential carryover effects from what they experienced in the larval environment. So we measured their susceptibility to BD, we measured their survival. So remember, we expected that as tadpoles' ponds dried out faster, we expected them to develop faster so that they could get out of those ponds. And it turns out, no, they did not do that, all right? So here we see days to metamorphosis on our y-axis, the proportion metamorphosing in our 3 drying treatments, constant in blue, medium in yellow, fast in red. And it was like, so like P equals close to 1, like not even a difference. All right. However, what we did see, and I'm not showing the data here, is that exposure to fast drying did decrease larval survival. So we had 3 to 5% surviving in our constant and medium dry treatments, whereas those tadpoles in our fast dry treatments, we had around 12% mortality there. Now, the other developmental characteristic that we looked at was size at metamorphosis. And again, drying had no effect. All right, so here we see days to metamorphosis on our x-axis, mass on our y-axis. Drying treatments, there was no difference. All right. However, we did see a slight, and I know it doesn't look that great, but I promise there is a significant positive effect here, where frogs with longer larval periods did tend to metamorphose a little bit larger. All right. So going back to our predictions, all right, we anticipated that drying would affect development, and that wasn't the case. All right. However, now we want to look at, are there potential carryover effects? All right. So again, what we looked at here was post-metamorphosis. We looked at susceptibility to BD infection as well as survival. So the way that we did this was after tadpoles metamorphosed, we brought them back in the lab. We're taking care of these little frogs under consistent lab conditions. And then once they were about 6 to 10 weeks post-metamorphosis, we started alternating weekly between exposing frogs to BD. So we would do that 1 week. The following week, we would swab them to quantify how much BD that they had. And we just kind of went back and forth out to day 63 post-exposure. All right. During this time, remember, it was a while before we got started with our experiment. And so we also continued to monitor the survival of frogs not exposed to BD, like just our controls in this study, as well as some extra frogs that we didn't even need in this study. We measured their survival up through 60 days post-metamorphosis. So let's take a look at our BD results first. And what we see here, I haven't even separated things out by drying treatment because there was absolutely no effect. What I did want to show you is that here we're looking at the timing after our first BD exposure. How many frogs had died? None. That was cool. Except with an infection study, maybe not. Maybe you do expect a little death. But we're looking at then our proportion of infected in this darker gray and uninfected in this lighter gray. And so what we see was that the proportion of individuals that were infected, that decreased. So that's indicating like there's some probably resistance developing within these frogs after they metamorphosed, but there was no effect of drying treatment on that. Where we did see a carryover effect was in survival after metamorphosis. And I want to emphasize that we have controlled for mass here. So what we see is timing post-metamorphosis going through 60 days and the survival probability of frogs reared as tadpoles under each of these 3 drying conditions. And what we saw was that drying did reduce survival. All right, so in summary, we anticipated that we would see developmental impacts of drying on tadpoles that we didn't quite see. We then expected potential carryover effects. Drying had no impact on susceptibility to BD infection, though it did decrease post-metamorphic survival. So what I want to tell you is that if you're interested in this, I have presented like the thinnest little slice of this massive, massive undertaking of a study. What I described, we actually did the same thing in 3 other locations using both the northern leopard frog and the southern leopard frog. We also looked at other aspects of their performance, including their antimicrobial peptides, their microbiomes. Like this was like, number one, imagine writing this paper. It was hard. Number 2, I feel so bad for the reviewers on this one because we had a lot packed in there. So if you want to learn more, this is where you can find everything from this study. All right. So keeping that in mind, I've gone through grad school at this point, studying climate change, mostly trematodes. I did do some BD work as part of my grad work. Then went to Pitt. We started looking at BD, started looking at pond drying effects on this disease. And now I want to talk a little bit about what I hope to get started at St. Bonaventure. So I'm planning on sticking with disease, but now starting to incorporate another thread besides just climate change and temperature, and maybe starting to look at how pollutants can impact disease processes. You'll notice that I'm using like planning and future words. And that is because I am 100% in my teaching era right now. So I'm in my 4th year. These are all the classes that I've taught so far. I literally have something new every year, and it's really hard. So my students have been very patient with me. We've been getting, you know, some small scale research done, but I'm currently, evolution is my last new one, I hope, for a while. So I think we'll be able to ramp up research a little bit after this year. So just to talk about kind of my inspiration behind this, I was kind of like an indoor cat during grad school. I did a lot of lab work. During my postdoc, I did a ton of field work. I know I didn't talk too much about it today, but I was out in the field a lot. And I was working at beautiful ponds like this, although when I saw them, it was like I could see this much of it through headlamp light. I didn't often get to visit my sites during the day, but this was a lucky day. And so it really got me thinking more about what else the amphibians are experiencing there. A lot of my field sites were near agricultural sites. And so I started thinking more and more about like pesticide input into these ponds. And so I started thinking about how that pesticide exposure might compound disease exposure for amphibians. And so what previous research has shown is that pesticides can reduce BD growth, both in culture and on amphibians. So what we're looking at here are results from Hanlon and Paris from 2012, where this was all done in vitro. All right, so they were growing BD, and I should mention that the infectious stage of BD is called a zoospore. So this is a measure of how many infectious organisms there are. And so what they did was that they grew the BD either just in regular media like they normally would, or with media with different pesticides added. And they tested carbaryl, which is an insecticide, glyphosate, which is an herbicide, and TM, which is a type of fungicide. And across the board, all of those reduced BD growth. All right. But we know that what happens in culture is not always what happens on an amphibian. And so the results of McMahon et al. In 2013 show that BD growth can also be reduced on tadpoles with pesticide exposure. In this study, they exposed the BD to an herbicide, atrazine, and then measured BD growth on tadpoles and saw that increasing concentrations of atrazine reduced BD growth on tadpoles. Now, sometimes when I explain this to incoming students in my lab, they think that I'm suggesting, let's just like pour pesticides into the ponds and this will help the frogs with their BD. No. Not what we're going for. But just to show that, that different pollutants can impact infection processes, and I think we need to understand more. So another thing that I want to mention here, this is work done by a group in our larger research group from when I was at Pitt. This comes out of a University of Nevada, Reno lab, where I'm talking about BD like it's one thing, there are so many different strains of this pathogen. And so what Sheets et al. Did was they looked at 5 different isolates of BD collected from different areas across the United States. So this one's Louisiana, New Mexico, Ohio, Tennessee, and Vermont. And they measured the temperature dependence, so temperatures on our x-axis, again, zoospores, that's that infectious part of BD. Zoospores per milliliter is on our y-axis. And they measured BD growth across this range of temperature for lots and lots of different isolates of this pathogen. And what I want to point out here, the main thing is that these strains, they're operating differently, all right, under temperature conditions. So this blue Louisiana strain, for example, it is outgrowing all of them. The others, New Mexico was kind of a weirdo, but the others tend to be grouping kind of down here. And interestingly, as part of my postdoc work, some of my other co-postdocs were looking and comparing some of these strains and how virulent they are to frogs in exposure studies. And not only did this Louisiana strain in blue outperform the others in culture, but it was also way more virulent than the Tennessee strand, what's one of the ones down here in red, in exposure trials with frogs. And so this has gotten me thinking, can these BD strains differ in other ways that we haven't quite tested yet? And in particular, are the ways that BD responds to pesticides, does it depend on strain? Because so far, what we've seen with previous studies, each of them has only tested one strain at a time, and we don't know. We don't know if it's different. All right. So I went a little faster than I expected, but that just leaves plenty of time for questions. I'd like to acknowledge everyone from all the labs I've been in. These are some of my undergrads I'm working with now, and many, many more people. And thank you for having me today. Mm-hmm. Right. So I'm just going to repeat the question. I know you can all hear me, but on Zoom they probably can't. So the question is about, like, in a study like this where there's amphibians and BD subjected to pesticides, are they tracking amphibian deaths as well? So This is prior research. This one's not mine. I think they were tracking that, and I can't remember off the top of my head. I think that survival was pretty consistent across the board. And that's not terribly surprising because so BD infects the skin of amphibians, except in tadpoles, it's a little bit different. So in tadpoles, BD is only infecting the mouth parts of them. And so it tends to be a much less severe infection. Unless it's so bad that the tadpole can't eat, normally survival is going to be pretty high in tadpoles. Yeah. Yeah, so the question is, when we found that, and I don't have the data showing here, this is all in culture, but when it was found that the Louisiana strain was more virulent, were we testing frogs from Louisiana? So that was the idea. We were going to do like this reciprocal transplant where that we're going to expose frogs raised in Tennessee and Louisiana, and from those locations, we'll expose them to BD from Tennessee and Louisiana, like a fully crossed design. And it was across the board. Louisiana BD was way more virulent than Tennessee. Yeah. Yeah. All right. So we're asking about geographic spread of the different strains. There's a lot. And it's like there's a lot of strains and a lot everywhere. So with BD genetically, in the United States, we have two main genetic backgrounds. They come from the global pandemic lineage, GPL 1 and 2. For example, at the field sites that I was working at in Tennessee, or not, when did I work in Tennessee? In Pennsylvania. Those field sites, we did send some of our samples out for genotyping. Some of our sites there, and I mean, they're separated by like, I don't know, they're all probably within 10 kilometers of each other. Some of the sites, it's like all GPL1, some it's all GPL2, some it's mixed. So it's either like a mix of the two or they're hybridizing. It's a mess. Yeah. Yeah, so we did, and that's something where it is sorry, I should repeat your question. So I'll repeat it so that people can hear. So here, like in the drying mesocosms, other things would likely change, like temperature, for example. And we did track temperature both like at the top of our water level and below. Our temperature like certainly increased with drying rate, and so in that way it did reflect what we would find in a natural system. Anything after metamorphosis, that was all consistent conditions at that point. Yeah, so the question is about, we've got this book, The Sixth Mass Extinction, which is certainly quite alarming. And the question is, based on my research, does anything that I've done make me contradict those alarmist views or kind of support those alarmist views? I would say a potential contradiction would be that, like, looking at like BD is bad. This fungal pathogen, it's bad for amphibians. However, at least in the United States, they seem to be doing well. And not from my own work, but from work coming with some collaborators, they've shown that amphibians have been adapting to deal with BD. So this is work by Jamie Voiles and Karie Richard-Zawacki. What they've shown is that over time, amphibian populations in Central America have recovered. And they've shown that recovery, it's not due to BD getting any less virulent, but it's due to the amphibians kind of ramping up their immune system. So I think that suggests a level of adaptation, and suggests some hope in all of this amid reports of sixth mass extinctions. On the other hand, one thing I try to stress to my students is that like we can't just rely on evolution to save us all. Like we have to do better in terms of what we as humans are doing. In terms of like with my system, minimizing disease spread is huge. Like the pet trade is one way that these pathogens get passed around. So there's a lot of room for improvement.
Thank you, Karie. You have more questions? We got class here at 14. You have more questions? Thank you very much.
Thank you.